Welcome all for the afternoon session of day one. So in this session, we have three talks by three laptops, followed by an education outreach program. So stay tuned. And before starting our first session, I would like to brief you all about the like how you can ask your question. So for the Zoom participants, please type your questions in the Q&A box of the Zoom. And for the YouTube participants, please type your questions in the YouTube comment section. So with this, I'll hand over to the first speaker.
Hello everyone. Welcome back to CCMB Open Week 2021. This session is brought to you by Rakesh Mishra Lab at CCMB.
DNA is the basis of life. It is the rule book of all creatures you see around, plants or animals. They're all controlled by information encoded within its DNA. The complete set of DNA that we inherit from our parents is called the genome. It is the subtle or gross difference in the information embedded in the genome that makes us different from one another. That is the cause for the variety in the living beings on this planet and its substrate for evolution. Any changes that occur in the DNA, then they are inherited and they make new species based on the selection process that appear time to time. All living beings inherit their DNA from their parents. And that is the continuity of the information that is acquired over the period of time during the process of evolution. Genome in a seed or genome in an egg has all the information needed to develop it into a plant or into an animal. How does this happen? How a seed develops into a tree or how an egg develops into an animal is a very interesting question that biologists have been uh, working on for several decades. And there are now lots of aspects of this complex process that we understand. Some of these interesting things we are going to tell you about. Since we know some part of the genome, what it does, we can make changes in that part of the genome. And if our knowledge is correct, then that part of the organism is going to be altered in the shape or the size or the identity. These are very exciting uh, discoveries and findings which we are going to show you today.
Hello everyone. Today we'll be introducing you to DNA, the thread of life. We all know that cells are the building blocks of life, and mostly DNA is the genetic material inherited from parents. This DNA is present in the cell nucleus. In humans, if we align all the DNA from all chromosomes end to end, the total length is about 2 meters long. And we have, in the human body, adult human body, we have approximately 40 trillion cells. Which means that the total DNA in the human body from all the cells is about 8 billion meters. This is a lot of DNA. It is the same length as taking about 500 trips from Earth to Sun. But how do you think is this 2-meter long DNA called the genome packaged in a very small nucleus? Does this long thread of DNA get tangled and messed up like the thread here? Or is there anything that can detangle it and neatly package it in the nucleus? Just like how we have several rooms in the house for different utilities, like we have a hall, which is mostly the sitting area, or we have the kitchen where we cook food, or we have the bedroom where we rest. The DNA is also neatly packaged and compartmentalized into the nucleus by the nuclear matrix, which is also known as the home of the genome. The smallest genomes that we know are mostly from prokaryotes like bacteria, which have about 3,000 coding genes. The smallest eukaryote, like the yeast, has about similar number of genes. But when it comes to higher organisms like worms or flies, we see that the amount of the genome increases, but the coding genes remain almost similar. When it comes to humans, there is a huge genome, which is about 3 billion bases, but we all have only 20,000 genes. We also know that human body has nearly 200 plus different cell types. A single cell called zygote gives rise to various organs during development, like the eye, the brain, your bones, lungs, stomach, the digestive system, liver, and the kidneys. All these organs are formed from the same DNA that was inherited from the parents. They all have the same DNA. So what is this DNA? So how do we know what this DNA is? The DNA is made up of 4 nucleotides called ATGC, a string of DNA here, as you see here. But how do we interpret this language? It's not like English where you can read words and sentences. Here, there are special proteins in the nucleus that can recognize the sequence and code the proteins. For example, say this is a gene that is required for making an enzyme that is required for digesting sugar. But it does not always mean that the DNA has to make enzymes or proteins. Some part of the genome is also non-coding. For example, all the DNA in all the cells in the brain have the same DNA as that of all the cells in the stomach. But how do we know that only the genes required for brain have to be expressed in the brain and the genes required for the stomach have to be expressed in the stomach? There are proteins called transcription factors that bind to non-coding DNA elements like enhancers and promoters and activate the genes that are required for the brain. These proteins are absent in the stomach, and hence this genes required for stomach are repressed. Now, how do these proteins know that they should not cross-activate? Like the genes required for stomach are not activated in the brain. These are restricted by DNA elements called boundaries, which are recognized by proteins called boundary factors. Imagine what would happen if all the genes that are required for digesting food are expressed in the brain. Do you think your brain would get digested? We all saw how important DNA was in making organs like brain and stomach. Does DNA also decide the body plan? We know that from small insects, fish, or mouse to a complex organism like human, they all have a similar body pattern where they have a head followed by a trunk and then the abdomen. What do you think would be the body plan deciding factor? To understand this, we need to understand a family of genes called Hox genes. Hox genes are responsible for body patterning. Hox genes were first discovered in Drosophila. There are several genes that are responsible for making the mouth parts and the head and contribute to the anterior part of the fly. There are specific genes that are required for forming the legs and the wings and are essential for the formation of the central region of the fly. There is a gene which is responsible for formation of the posterior region of the fly. So if this is a normal fly and if we mess with this gene that is required for formation of a second pair of legs, this is something that we usually do in the lab. What we get is that now when this gene is messed with, the mutant flies will have legs growing out of their head. Similarly, we have other genes in the Hox system. Like the ultrabithorax. When this gene is messed with or mutated, we see that if a normal fly has one pair of wings, the fly with, one second. The fly with ultrabithorax mutation will now have two pairs of wings. Similarly, we also know that if we mess with the genes that are responsible for the posterior region of the fly, we can see here that if the normal fly has a specific pattern, the fly in which the gene abdominal B is mutated gives different patterns. Nature has already done a lot of tinkering with the DNA, and we see that in nature we have flies which have two wings, we have flies which have one wing, and we have flies or insects that have different types of abdominal patterns. So what do you think would happen if there is a Hox mutation in the human? Do you think there would be human beings with 4 hands? Actually, it is true that if Hox genes are mutated in humans, we do see babies born with multiple hands. This is a drastic scenario but small changes in Hox genes cause usual phenotypes where we all would have seen people who have multiple fingers or extra fingers. Like in this case, there is an extra little finger and we have an extra thumb. It also causes extra toes. Mutation in Hox genes also cause extra toes. We see people with 2 toes, 3 toes, and 4 toes. This is very similar to what we see in nature in different animals. We have one-toed animal, two-toed, three-toed, and four-toed animals. So nature has done a lot of tinkering with their DNA over the span of evolution to create diversity that we now see on Earth. Following this, we have a quiz. This is the diversity we're talking about. We have insects which have multiple number of legs. We have insects that can creep, that can crawl into crevices. We have insects that are limbless, and we have insects of different body shapes. And we have organisms that can live on earth, water, air, etc. Now is the quiz time. So log into Mentimeter and use the code displayed here, 83705896, to take the quiz.
So now we are starting the quiz. First question is, ask me. So to join the quiz, the password is 83705896. Share the screen. So our first question is,
Today share conference one. This is done. One second.
what is the thread of light? Fast replies will got more points here. So reply fast. Your time is up. And the DNA is the thread of life. Is the right answer. Where is DNA found in the cell? Reply fast. Options are nucleus, cytoplasm, and cell membrane. And the right answer is nucleus. DNA is found in the nucleus. Our second question, Lakshita gave the fastest answer. So now we move on to the next question. How long is the human DNA? Our options are 60 meter, 2 meter, and 20 meters. Meter is the right answer. And there are many who gave 60 meters also. Just to clear your doubt, it was for per cell DNA, not for the whole. Again, Lakshita gave the fastest answer. Now we have next question. What is the home of the genome? Options we have is R cell, nuclear matrix, and nucleus. Reply fast to get more points, guys. And nuclear matrix is the right answer. Gave fastest answer in this session. Moving on to the next question. Do all the cells in our body have the same DNA? As options we have are yes, no, or maybe. So answer is yes. All the cells in our body have same DNA in particular person. So the answer is yes. We get the fastest answer and move to the next question. What does the non-coding DNA do? Options are nothing, regulation, codes for protein. And answer is the non-coding DNA regulate the expression of different genes in different cell types. So they do not code for protein, but they play important role in regulation of genes. Move on to the next question. How many major alphabets are there in the DNA? Options are 3, 4, and 5. The right answer is 4, not 3. We have A, T, G, and C in DNA. So till now, Ayush Goyal is leading the scoreboard. Move on to the next question and next and last. What are the sets of genes that decides body plan of organisms? Options we have SOX, FOX, and FOX. The right answer is FOX genes. And we have a winner here, which is Ayush Goyal, winner for the quiz. So now we'll take question answers for the whole session. And I will give speaker to
Hello. So, are you able to hear me? So one of the questions that was asked was, we inherit DNA from our parents, and even our parents must have inherited from their parents. That means, do we have the genes of our ancestors passing through? Yes, you will. Like, for example, take my example. I get about 23 chromosomes from my mother and 23 chromosomes from my father. But take my father. My father is going to get 23 from his mother and 23 from his father. But I got only 23 from him. So as you calculate, eventually, you are more related to your parents than to your great grandfather or your ancestors earlier to them. So the similarity kind of decreases as you go towards your ancestors. The next question was, how many nucleosomes together form a chromosome? So a nucleosome kind of wraps around 147 base pairs, and the amount of DNA that is there in an organism changes. Like a yeast has something like 12 MB, which is 12 into 10 power 6 base pairs, whereas humans have something like 3 into 10 power 9 base pairs. So that's a lot of DNA. Because the amount of DNA changes, the number of nucleosomes also change depending on the chromosomes. The next question is, what are Hox genes? Now, for example, I have my hands closer to my head and my legs closer to my stomach, and not the other way around. Like maybe my hands are closer to my stomach and legs are on my head. That's not that doesn't happen because Hox genes kind of regulate where your hands have to be, where your legs have to be, and where exactly your different internal organs like liver, heart have to be positioned. The next question that is related to Hox genes is that we have seen that a mutation in Hox genes causes extra finger like what Hrithik Roshan has. It's not a disease, it's just an inconvenience. You wouldn't have a problem, it's just that you have a mutation in one of the Hox genes, right? Now, animals and insects, do animals and insects have DNA? Yes, all living organisms have DNA. It's just that some of the viruses might use RNA instead of DNA as their genetic material. That's one of the exceptions. Now, another question was why some fly have 2 wings and some fly have 4 wings. Okay. A fly has 2 wings normally, and there are 2 organs called balancers just below the wings, which help in balancing. Normally, a gene called UBX is responsible for wing formation. This is normally inactivated in one of the segment where you have balancers. But if it gets activated there, then you will have 4 wings instead of 2 wings. Okay. Another question is, what is recombinant DNA technology? So it's a way of changing DNA. Now there are other techniques called genome editing where you can edit your genome. If you have diseases, for example, there has been a new trial that has been going on in FDA where HIV is trying to be modified using CRISPR. So these kind of gene editing can be done, but this has to be regulated because people might be using this technology to kind of have babies who have a different eye color or a different kind of hair. So that has to be regulated. But for disease purposes, this can be a very boon for people who are suffering. So is it possible to replace the mutant gene with normal while doing germline therapy? Yes, it is possible, but it's not yet approved. Another question that people had was, does every drop of blood have DNA? Yeah, your blood consists of different kind of cells like RBC, WBC. So WBCs and platelets do have DNA in them. Sorry, WBCs have DNA in them, whereas some of the cells also secrete DNA into the blood. So there is blood outside also, there is blood within the cells also. So every drop of blood does have a lot of DNA. If there are any further questions, you can raise your hand or we'll stop. Okay. Can you please uh, ask your questions in the Q&A itself? There's a question which says if we add in human genome and animal DNA, which last for many years, then the animal can also get that characteristics. It's a good question. You could actually, it's the other way around that we do in our labs. We take kind of take genes from different organisms and transfer into different organisms and kind of see what functions do they have. But it's difficult only by changing a single DNA or a gene. You will not have all the characteristics, but you could have certain functions that could come because of that gene. So please tell about Human Genome Project. Okay. So what happened was, like we were saying that the DNA composes of alphabets like A, T, G, and C. Each person has a different combination of these 4 alphabets to make up their DNA. And your DNA sequence, which is the combination of ATGCs, is going to differ from your sister's ATGCs combination and also from your parents. So people didn't know in 95 and in 90s what the sequence of the human genome was. And then there was a human genome sequencing technology that was genome sequencing technology that was developed, which they had used to kind of figure out what is the content of the genome, what are these ATGCs that are there in a particular human being. So that kind of completed in in a lot of years and a lot of money back then. But now you could get your genome sequenced in $1,000 and in probably 2 days or a week. If a mutation is detected in the embryo stage itself in an organism, is it possible to repair it? Yeah, you could repair. With that, we'll close the Q&A session. Thank you for your attention.